Good morning, children. Today in this session, we will continue with the subject enrichment activity. So in this video, we will discuss about the experiment, the release of carbon dioxide during respiration. Aim: to show experimentally that carbon dioxide is given out during respiration. Theory: all living things shows respiration. It is a chemical process that occurs inside the cell, hence it is called as cellular respiration. Respiration involves the breaking down of food to release the energy and carbon dioxide. It is a reaction reverse to that of photosynthesis. So you can see the equation of respiration C6H12O6, that is glucose. It reacts with the oxygen and results in the formation of carbon dioxide, water and energy. So the raw materials for respiration are glucose and oxygen, and the end products of respiration are carbon dioxide, water and energy. So if you observe carefully, the respiration is nothing but the reverse of photosynthesis. There are two types of respiration in animals: aerobic respiration and anaerobic respiration. Aerobic respiration needs oxygen and anaerobic respiration occurs in the absence of oxygen. There are three pathways of respiration as shown below. Glucose molecule, that is a 6-carbon molecule, in cytoplasm it will broken down into 2 molecules of pyruvate. So pyruvate is a 3-carbon molecule and it releases little amount of energy. And the pyruvate molecule, in the presence of oxygen or in the absence of oxygen, it undergoes different modes of respiration. So in the presence of oxygen, that too in the mitochondria, the pyruvate molecule will be converted into carbon dioxide, water and energy will be released in the form of ATP. Whereas in the lack of oxygen, that too in the muscle cells, the pyruvate molecules are converted into lactic acid, which is a 3-carbon molecule and releases little amount of energy. In the absence of oxygen, in yeast cells, the pyruvate molecule will be broken down into ethanol. It is a 2-carbon molecule and carbon dioxide and energy is released. So the respiration in the yeast is called as anaerobic respiration. Why? Because it takes place in the absence of oxygen. The energy released in cellular respiration is immediately used to synthesize a molecule called ATP. Plants also release carbon dioxide during respiration. The exchange of gases during respiration takes place through small pores present on the leaf called as stomata. Carbon dioxide can be tested by lime water test. A freshly prepared lime water is calcium hydroxide. When carbon dioxide is allowed to pass through it, an insoluble compound called calcium carbonate is formed, which makes the lime water milky. We can see the chemical equation: calcium hydroxide, that is lime water. When it reacts with carbon dioxide, it results in the formation of calcium carbonate. So it forms a milky precipitate. Along with the calcium carbonate, it releases the water molecule. Test for release of carbon dioxide during respiration in animals. Materials required: 2 test tubes, a cork with 2 holes, 2 glass tubes, syringe and lime water. So let us observe the procedure.
To show experimentally that carbon dioxide is given out during respiration. Respiration in animals. Materials required: freshly prepared lime water in a test tube, cork with 2 holes, glass tubings bent at 90 degrees. Procedure: take half-filled test tube with freshly prepared clear lime water. Fix the cork in the mouth of the test tube. Fix the glass tubings A and B in the cork such that end of glass tubing A is dipping in lime water. Put your mouth at the end of the tube A and exhale out air with full force. Pass exhaled air vigorously for at least 1 minute. Observations: You observe as the exhaled air bubbles pass through lime water, it gradually turns lime water milky. Conclusions. As the clear lime water turns milky only with carbon dioxide, therefore, we can say that carbon dioxide is present in the exhaled air and is produced by the process of respiration.
Procedure. Take some freshly prepared lime water in 2 test tubes. Fit cork with 2 holes in test tube A and B. Fix 2 glass tubes in this cork of test tube A as shown in the figure. Exhale air into the tube and record your observation. So in another test tube B, which has the lime water, pass air through syringe and record your observation. So in the setup A, exhaled air has to be passed through the test tube A, whereas in the case of test tube B, the air has to be passed through the syringe, and record your observation. Observation. In test tube A, the lime water turns milky sooner compared to the test tube A. Conclusion. The exhaled air contains lot of carbon dioxide which turns lime water milky. This proves that during respiration we exhale carbon dioxide gas. Precautions. The glass tube should be dipped into the lime water. The lime water should be freshly prepared. To test the carbon dioxide release by plants during respiration. Materials required: conical flask, small test tube, cork, thread, germinating seeds, a bent tube, a beaker, water and freshly prepared lime water. Procedure. So here is the video. Observe the procedure carefully.
Respiration in plants. Materials required: germinating gram seeds, dropper, conical flask, cotton thread, glass tubing bent twice at right angles, small test tube, cork with 1 hole, potassium hydroxide solution, 100 cc beaker half filled with water, Vaseline. Procedure. Place about 50 germinating gram seeds in a conical flask. Tie a cotton thread to the neck of small test tube. Pour about 4 ml of KOH solution in it and suspend it in the conical flask. Potassium hydroxide should be freshly prepared and concentrated. Close the mouth of conical flask with a cork in which a delivery tube is fitted, such that thread tied to the small test tube is held firmly. Fix the one end of twice bent glass tube in the cork and the other end is dipped in a beaker containing water. Apply Vaseline on the cork so as to make the apparatus airtight. Leave the apparatus undisturbed for 2 hours. Observations. After 2 hours, the level of water in the delivery tube rises up as compared to level of water in the beaker. Conclusions. The rise in level of water in the delivery tube indirectly proves that germinating seeds produce carbon dioxide. The carbon dioxide produced by germinating seeds is absorbed by the potassium hydroxide solution. Now, as carbon dioxide is produced due to the consumption of oxygen during respiration, therefore, pressure of air within the flask falls. To make up for this loss in pressure, the air from outside exerts pressure and hence forces up the water within the delivery tube. Procedure. Take 2 conical flasks and add germinating seeds with a little water sprinkled over it. Fix the mouth of the conical flask with cork in which a bent tube is fixed. Suspend a small test tube containing potassium hydroxide solution in it with the help of a thread in a conical flask A. Allow the mouth of the bent tube to be immersed in the water in setup A.
and lime water in the setup B as shown in the figure. Observe the setup A and B carefully. Setup A contains a small test tube with potassium hydroxide solution, which is not there in the setup B. And if you observe the beaker containing liquid, in the setup A, the beaker contains water, and in the case of setup B, the beaker contains lime water. Observation. In setup A, the water level in the vent tube dipped in a beaker increases after a few hours. This is because the oxygen present in the conical flask is taken up by the germinating seeds and carbon dioxide is released due to respiration, which is absorbed by potassium hydroxide solution present in a small tube. Hence, air pressure in the flask reduces and water level rises. Whereas in the case of setup B, freshly prepared lime water turns milky. This is due to the excess carbon dioxide released in the test tube during respiration of germinating seeds. Conclusion. This shows that carbon dioxide is given out during respiration. Precautions. Lime water should be freshly prepared. Potassium hydroxide solution should be freshly prepared. To demonstrate the production of carbon dioxide during anaerobic respiration. Theory. During anaerobic respiration, the food material, that is glucose molecule, breaks down into carbon dioxide and alcohol in the absence of oxygen by the microorganisms such as yeast. This type of respiration is called as anaerobic respiration. Materials required. Sugar solution, concentration should be approximately 15 to 20%. Test tubes, delivery tube, 1 hurled cork, freshly prepared lime water, petroleum jelly or Vaseline, another one is yeast powder. Procedure. Observe the video carefully to get the clear picture about the procedure. Experiment to demonstrate the anaerobic respiration in yeast. In anaerobic respiration, glucose molecule, that is C6H12O6, will break down into carbon dioxide, ethanol and energy will be released. Anaerobic respiration is the process of producing cellular energy in the absence of oxygen. During anaerobic respiration, glucose molecules are broken down into carbon dioxide, ethanol and releases energy. Materials required. Yeast powder, flask, glucose powder, liquid paraffin, lime water, delivery tube, thermometer. Prepare glucose solution in a beaker. Take the fresh water in a beaker and add glucose powder into it and stir it with the help of a rod so that the glucose will be completely dissolved in the water. Remove the dissolved oxygen from the glucose solution by heating it for a minute and cool it without shaking. So after boiling, you can observe air bubble formation. It clearly indicates that oxygen is removed from the glucose solution. Pour the glucose solution into the flask. And add some yeast powder to the glucose solution. Pour some liquid paraffin. If it forms as a layer on the glucose solution, it prevents the entering of oxygen. Close the flask with 2 hurled cork. take the lime water in a test tube and keep it in the test tube stand. Arrange a delivery tube from the glucose solution to the lime water through one hole of the cork. You can see one end of the delivery tube is present in the flask and one of the end of the delivery tube is present in the lime water. Insert thermometer into the glucose solution through another hole. Record the initial temperature of the thermometer. Observe carefully, note the initial temperature. It was around 33.5 degrees Celsius. You can see lime water is clear. So before the experiment, the initial temperature is 33.5 and the lime water is clear. You can see the entire setup. You can observe thermometer, 2-hole cork, flask, liquid paraffin, glucose solution with yeast, delivery tube, lime water. After 1 day, temperature is increased in the thermometer. Record the final temperature. So it is 36 degrees Celsius. So from this we can say that in anaerobic respiration in yeast, energy is released in the form of heat. Lime water turns into milky white due to the release of carbon dioxide in anaerobic respiration. The alcoholic smell comes out from the flask. So from this we can say that the end products of anaerobic respiration are ethanol, carbon dioxide, and energy is released in the form of heat. Procedure. Fill the test tube completely with sugar solution. Dissolve some granules of yeast in warm water in a test tube and mix it well. Add about 20 drops of yeast solution or yeast powder into the glucose solution. Using petroleum jelly, fix a cork tightly with the delivery tube bent twice at a right angle. Place the another end of delivery tube into the test tube containing freshly prepared lime water. Observe what changes are caused in the color of the lime water with time. Observation. The color of the lime water becomes milky due to the carbon dioxide released during anaerobic respiration by yeast. We will continue further in next session. Thank you.